Hello, welcome to this video abstract. My name is Taeseok Daniel Yang, and I'm PhD student at Center for Cell Dynamics, Korea University. I'm studying biological mechanism behind the various self-organizing patterns in biological systems. Here, I'd like to introduce our new NJP article entitled Trail Networks Formed by Populations of Immune Cells. This work includes experimental observation on remarkable complex trail network patterns formed by population of crawling red microglia. Microglia are a type of immune cell, and they are the resident macrophages acting as the main form of active immune defense in the central nervous system. A general feature of immune cells is perhaps a good motility, and it is also true for our MG cell. Interestingly, crawling MG cells are neither purely diffusive or ballistic, showing a long-range directional persistence. This is a close-up view of a freely crawling MG cell. Surprisingly, they prefer to make zigzag turns in crawling, and this property is closely related to their directional persistence. This phenomenon was discussed in our previous report, and in this work, we are more interested in their collective population dynamics. The cell population show a very complex network patterns of migratory trail, and the overlaid stack of images on the right-hand side clearly reveal the patterns. MG cells are harvested from the rat brain and placed on the culture dishes, and their behaviors are monitored by time-lapse imaging over several days. Typical cell density is about 200 cells per square millimeter. The cells seem to leave some chemical marker or some substrate-bound attractant behind their passage, similar to ants leaving pheromones or amoeba excreting CAMP. Then the attractant from MG cell guide the cell to change their moving direction toward the existing passage. Sometimes the cell escape from the track and rejoin into another track. In fact, we found that MG cells leave a very faint trace of integrin alpha 5, which is known as an adhesive molecule. They also lay down some fragments of needle-like structures having a diameter of tens of nanometers. We believe that cells are attracted to these physical as well as chemical cues. Based on these findings, we built a mathematical model cell population and could reproduce many key features of the observed trail patterns. Freely crawling model cells have a directional persistence with a zigzag turning preference, much like the microglia. And the model cells are assumed to lay out the attractant that is not diffusing but slowly decomposing in time. The observed patterns are well reproduced by populations of model cells interacting with each other. The right-hand side shows a close-up view of a few cells recognizing each other's trail. Moreover, the network evolved slowly in time. Some trails were enhanced or dually created, while some that were rarely visited disappeared. The haptotaxis mediate evolving networks, which are neither random nor regular, define a new class of self-organized patterns formed by populations biological cells. The trail network, as compared with the random network, seem to confer a more efficient traffic for the brain immune cells. Summary: haptotaxis-mediated interacting microglia cell population can self-organize to form complex network of trails and reproduced by mathematical model cells interacting with each other through a chemical attractant. These networks formed by cell-to-cell interaction are more efficient than random networks. Thank you for your attention.